Alright guys, so this video is a closer look into the adaptive immune system, which works with the more general and non-specific innate immune system in the body's overall immune system. And the body's overall immune system is really a collection of the natural defense mechanisms to defend the body against infections brought on by the pathogens. The adaptive immune system is more specific and advanced and thus more effective, and we will be focusing on the antibody-mediated response and the cell-mediated response. So the antibody-mediated response includes the B cells, and B cells are white blood cells that are created and mature in the bone marrow. They become activated and respond when the antigens bind to their antigen receptor proteins on their cell surfaces. Now, we have some new vocab terms. An antigen is really something that stimulates B or T cells, and this includes like parts of pathogens. Antigen receptors bind to the epitope of antigen, which are just small accessible parts. Now, the B cells and T cells display specificity in the sense that all the antigen receptors on one cell is completely different and unique when you compare it to those antigen receptors on another cell. In other words, each cell responds to a different antigen. Now if we look at this diagram, we can see a B cell right here, and if we zoom in on the antigen receptor, we can see that it consists of light chains and heavy chains, and they are combined by disulfide bridges, and they have two antigen binding sites. Now when a B cell is stimulated, it's going to have a phosphorylation cascade, and that's going to lead to the production of a protein called antibodies, and these antibodies are classified as an immunoglobulin. And these antibodies bind to antigens that are fixed on pathogens or freely float in the bloodstream. They actually do most of the defending rather than the actual B cells. We have to know that antibodies are also specific, as different antibodies bind to different antigens, and the type of antibody you get is determined from the plasma B cell that creates it. Now, antibodies don't actually destroy and kill pathogens, but they do inactivate it or inhibit their functions. And they can do these in three different ways. Neutralization is when the antibodies all work together to bind to the proteins on antigens, and this disables and inhibits the antigens from binding to cells and infecting them. Opsonization is when the antibodies bind to the proteins on antigens, and they create this structure which is recognizable to the neutrophils and macrophages for phagocytosis. And through the activation of complement system and pore formation, complement molecules can bind to the antigen-antibody complexes, and the membrane attack complex forms will then go on to lyse the foreign cell or invite more macrophages and neutrophils to consume the foreign invader through phagocytosis. Now, B cells are activated when they bind to pathogens via their antigen receptors or antibodies, and they display the antigen fragments that they collect on their surface with the help of a class II MHC protein. We're going to talk about this later in the video. We should know that antigen fragments are specific to the B cell. Now, an activated helper T cell is going to come in and it's going to direct a cell-to-cell contact with the B cell. This is going to stimulate a phosphorylation cascade within the helper T cell, and cytokines will be released from the helper T cell to the B cell. Cytokines will bind to the receptors on the B cell, and this is going to create a phosphorylation cascade within the B cell that then leads to proliferation and differentiation, so that B cells create many more memory B cells and plasma B cells. Memory B cells are the ones with the antigen receptors and really work with the activated helper T cells to create the cycle onwards, while the plasma B cells are the ones that produce the specific antibodies. Now, in the cell-mediated response, we have T cells, and like the B cells, they originate in the bone marrow, but later they migrate to mature in the thymus, hence the name T cells. The antigen binding receptors are different to those of B cells, and if you were to look in this diagram, you'd see that they consist of an alpha chain and a beta chain, which are bridged together by disulfide bridge, and they have one antigen binding site. Now, T cells bind only to antigen fragments that are present on the surfaces of cells through something called antigen presentation. And cells that participate in antigen presentation use MHC proteins, or the major histocompatibility complex proteins. And here you can see T cells binding to these antigen fragments that cells put out with the help of MHC proteins. There are two types of T cells, helper T cells and cytotoxic T cells. Now, helper T cells, their main goal or function really is to trigger the humoral / antibody and cell-mediated response, and doesn't really carry them out. So when a helper T cell binds to the antigen fragments of a cell, for example, B cells, macrophages, dendritic cells, infected host cells, they're going to bind to those antigen fragments and the class II MHC molecule with the help of CD4 accessory proteins. And this binding stimulates a phosphorylation cascade within the helper T cell that stimulates the release of cytokines, which triggers the humoral and cell-mediated response. This release of cytokines also activates other helper T cells, which really helps further the immune response. Now, cytotoxic T cells actually take a more active role in destroying these infected cells. They use toxic proteins to kill infected cells before the pathogens really reproduce and grow. And they become activated when helper T cells really sends out the cytokines to activate them. Cytotoxic T cells recognize class I MHC molecules, and they bind to the antigen fragments and the MHC1 proteins with the help of an accessory protein called CD8. This binding triggers a phosphorylation cascade in the cytotoxic T cell, which releases chemicals like perforin. And these chemicals bind to the receptors of infected cells, and this leads to a phosphorylation cascade that ultimately leads to the apoptosis of the infected cell. The dead cell remains are left by the cytotoxic T cells, and the antibodies come to pick up the mess. And now, an important part of the immune system is the immune memory. And it's really important because this allows for a long-term protection after overcoming and defeating a disease. And really, an immune response becomes much faster after an initial primary immune response. When the cells are still learning how to respond, it takes them time. And once they finally do this, they gain the right, I guess, quote-unquote, knowledge about how to counteract this infection. And when a secondary immune response comes in, the response is going to be much stronger, much faster, and it's going to have a more prolonged effect, all thanks to the cells knowing how to respond and this reservoir of B and T cells that were produced from the initial primary immune response. These cells have long lifespans, and they'll participate in cell division and continue to grow. As you can see from this chart, you can see that during a primary immune response, the cells are going to take a longer time and produce fewer antibodies, but with the secondary immune response, they're going to produce more antibodies for stronger effect. And here you can see B cells reproducing and maintaining their presence, so just in case that same antigen does come back. Now, you should be able to understand what the adaptive immune system is and what it really consists of. And here's a diagram that really summarizes everything I've talked about. And using your knowledge, you should be able to interpret this diagram and really be familiar with the adaptive immune response. Thank you for watching this video.